turn on and off. It's important to understand because that's a big part of how these stem cells are going to specialize. Stem cells will activate certain genes in the process of differentiating into different types of cells. Transcription factors are major key players here. They're typically, but not always, proteins, and they determine which areas of the DNA code will get transcribed into mRNA. That mRNA can eventually be used to make specific proteins that can impact what a cell is going to look like and what a cell is going to do. That means transcription factors have a major role in determining which genes are expressed in a cell. Because a cell that's going to become a skin cell is going to have different areas of genes expressed than a cell that's going to become a stomach cell. There are both internal and external cues for stem cells, which can involve these transcription factors. Examples? Okay, an example of an internal cue could be transcription factors present in the cytoplasm of the original starting zygote cell, which will eventually be present in the cells that come from it. Now, the specific location of the stem cell within the developing embryo can matter because the transcription factors available in different areas of the developing embryo can differ in quantity and type, which could impact what a stem cell differentiates into. External cues could involve cell signaling from other cells next to it. External cues can even be environmental effects like temperature. There's still a lot of research in this area, and we can't wait to see what scientists discover about this in the next decade.
Okay, so they start talking kind of over our heads because we don't really know what transcription is yet, and you will, and it will make a lot more sense. And that's why that with stem cells, when we're talking about those, we talk about the begin later. So that way we can apply that to them later after we've understood it. So cell signaling, and we are going to talk about that today. So what was the first question?
What is cell specialization?
All right, so who wants that one? What is cell specialization? All right.
Cells in an organism can develop in different ways to perform different tasks.
There you go. So if you are unicellular, do you really have cell specialization?
No.
No, you're just one cell. You're just one. Like a lot of the protists you were watching swim across your field in the microscope. I mean, that's just a one cell. Your whole cell is your body. But if you're multicellular and your body wants to be able to do many things simultaneously, you've got to have cell specialization. So that's it. Okay, number 2, what type of cells are the undifferentiated blank slates? Waverly. And then Mara, you do the last one. Stem cells. That's it. So tell me, what have you heard about stem cells? Or what do you know about stem cells? Has everybody heard of stem cells before? Stem cell research or anything that you know about stem cells?
I feel like it goes in your body or something if they're not working or something's not working.
So they can use in some type of, it's called regenerative medicine. You can use them, see if but you have to activate them and they can become any type of cell, right? I mean, think about it. If you've got stem cells in your body now, even though you're not an embryo anymore, but you have adult stem cells. Some of those are in your bone marrow, and that's why you make new kinds of cells. Your blood cells, your red blood cells, your white blood cells, they come from those. But there's also, they're located in other parts of your body. So it'd be a pretty cool thing if you could insert stem cells in order to become a new healthy type of tissue to fix a problem. That's where a lot of the medicine's going to go, which is pretty amazing. Okay, Amara, how about the last one? What causes one of those cells to start becoming a new, like a type of cell? Okay, so there's something that stimulates something to cut on. So it could be a transcription factor is going to cause it. They said an external cue is going to cause it. But in order, what has to happen to the DNA that causes that cell all of a sudden start to become something new?
Something triggers it or an instruction?
All those things are going to trigger what to happen. I thought they said it maybe. Gene activation. That's it. So there's a transcription factor, which we'll find out there's some faces to those that we'll put with, what that means. So the gene has to cut on either by transcription factor or an internal cue, they call them, or an external cue that can cause that. So here's an example of how it can affect development. In turtles, temperature determines sex. It determines gender. So if the temperature in that developing egg is really cold, there's your external cue, it will cause that little embryo to develop into a boy. And if it's warm, it'll develop into a girl. So think about it, that can happen with us. And so it doesn't matter what the predestination is. It's the fact that the temperature external cue triggers a response, and that turns on these genes, and they become male or female.
How does that work with us, though? How does that work with us? Like, how does
it doesn't. I say that, and it doesn't. We have X and Y chromosomes. So you will get whichever chromosomes that predestine you to be a certain gender. However, there's genes on those chromosomes. So those genes can determine all of your secondary sex characteristics, you becoming fully male or fully female. And so that's where genes cut on, genes cut off. And so if you don't have a specific gene there, maybe a mutation in a gene, that could cause a problem in that development. Or the gene doesn't cut on, something prevents that. So it's all about gene switches. So if you think about, you have 20,000 gene switches in every single one of the nuclei of your cells. You have the exact same DNA in every single cell, because you only got one copy when you were made. So the same gene. So if I could trigger a leg-making gene in the skin cell, I could start to form a leg.
out of your own?
So they've done it in college. I saw this was, they're just starting that when I was way back in college. But there was a gene that got cut on in the eye of an insect. And the insect, it was the leg-making gene, and the leg started forming out. So if you can trigger the gene, so think about the implications when it comes to like cancer. If cancer are your own cells that have gone rogue, that a mutation has caused this proliferation of cells, what if we could go in and cut off that gene in those cells to prevent them from reproducing? So it's going to happen in your generation. We're going to see more and more effective treatments for cancer. Hopefully in mine too, but definitely in yours and your kids. There's not going to be one cure, but there will be multiple treatments that will hopefully help to not annihilate a disease, but at least get it to manageable numbers. So, which will be great. So that's pretty much it. So usually folks have a question between specialization and differentiation. So look at your vocabulary words and let's see. So we heard specialization. So what is differentiation again? Jalecia, what do you have for that? I don't even know what number it was. I just
for that one I put is the process by which a less specialized cell becomes a more specialized cell type.
Right. So it's becoming a cell type. Differentiation. That's all that means. I'm pulling them up. I have them right here. So that's number 2. All right. Stem cell, we already said, is a blank slate, undifferentiated cell, either of an embryo or they're found in adults also. So we're going to see a couple of video clips that go along with that breakout activity. So it's actually in the packet, the front part of it. So I think this will trigger a lot of, you'll see the application of it, but also a lot of the conversation. You'll be able to see some of these. All right, xylem cell. Now all this next little run from 4 to 9 are all specialized cells. In fact, 1, 2, 3, 4, 5 of those are in the breakout activity. Okay. All right. So how about a xylem cell? That was one of them that was on there. Let's see. Rocio, how about you?
It's a petition, a vascular plant that transports water and some nutrients from the roots to the leaves.
Okay, so if it transports water, it's got to be able to get the water through pretty quickly. So think about that. And so when you look at the pictures, that might help you figure out which one it is. All right, let's see. Everybody's gone, right? Okay, so let's go back to Karen. How about phloem cell?
Vascular tissue in plants that conduct sugar and other amino solid products down rich mediums.
So phloem and xylem, both types of vascular tissue. We're gonna find that those really are like the blood of a plant. Have you ever like picked a stem of a plant or something that's like this gushy, like stuff that comes out of it's like clear? Or like, have you ever seen aloe and you break off the stem of aloe and it's the aloe itself? All that's vascular tissue. So xylem and phloem's in there. And it runs through the vein of a leaf, just like our blood runs through our veins. Excuse me. All right, epithelial. Amari, what'd you get for that?
Composed of one or more layers of densely packed cells.
Of densely packed what?
Cells.
Cells. Okay, so epithelial line and protect. So your cheek cells were epithelial cells. Okay, your skin epithelial. Anything that lines your throat, your esophagus. I mean, all that. Any of the liners and any of the protectors. Okay, how about neuron, Kenzie?
It carries electrical impulses.
So that's a fancy word for what kind of cell? Nerve. So it's branched. So we know that it has all these extensions, so it can pull information in. All right. Waverly, how about leukocyte?
Cell in the body, fluids involved in contracting foreign substances and diseases.
So that's a fancy word for what kind of cell? White blood cell, RBCs. Now, there are many kinds of white blood cells. Many kinds. It's a whole army. It's like the first line, then there's the second line, then there's the third line. And it is, you could do a really cool analogy with the military and your immune system. It would be a really neat thing because it's pretty amazing how they all have these specific jobs. All right, so Jaleesa, how about erythrocyte?
They are red blood cells that travel in the blood. They're like another word for red blood cells as well, since you were like saying, I don't remember, when was it, like Wednesday, I think you were mentioning that there were going to be a word that on there that we like talked over already. So like that word.
Okay, good. So Rocio, harder question. You remember the protein that's inside of red blood cells that carries oxygen and carbon dioxide? Starts with an H. Put you on the spot.
I don't know. I want to say hydrogen, but I don't think that's right. So
It has hydrogen in it because it's protein, because it has CHON. There's a name. I don't know who I saw first. So, all right, one of you girls, I saw one of your hands pop up. I just didn't see.
I'm not sure if I know what it is exactly.
All right. Hemoglobin. Hemoglobin. So hemoglobin is that protein. It has a lot of iron in it. So if you're anemic, you have low iron, and that means you can't carry oxygen and CO2 as efficiently as somebody anemic.
Yes.
She's anemic.
That vegan lifestyle. Girl.
Exactly. Thank you.
It doesn't have to do with that. You know, I
Yes, it
still
does.
eat meat. I still have terrible iron.
Yes.
Sometimes it can also be genetic. It really can. It can be genetic. And it can also be the size of your red blood cells. So Waverly could have smaller red blood cells. Just don't have as much hemoglobin. Can't carry as much oxygen, you know? I don't know. Some people battle that along. I mean, they just do. All right. Let's see. Next one. I guess that's it with those. Yeah. All right. Let's move on then. So in your packet, that last sheet. And let's look at the front because it has the back. It should be the crossword puzzle. Well, I said it's the last sheet.
Oh, this is the.
Not the new one, but the old one.
This is the question right now. God bless you.
Yes, ma'am.
Have you ever met somebody who had like an enlarged like major organ, like one of their major organs is enlarged?
I don't think so. I know that it happens, but I don't think so.
I mean, it's like, is it like deadly?
Oh, yeah. It can be. It can be, especially the heart. You know, that's what can cause heart attacks and like premature heart attacks and stuff in athletes.
Oh,
Well, that's one cause of heart, heart
my
problems.
mom. The left side of her heart is enlarged.
So what are they doing about it?
Like, it's been a long time, but they haven't done anything.
Looking for my copy. I might have already put it up. Okay, let me pull this up. It's on the front. Let's see. I need to make sure that I type in the right URL because that's not quite right. All right, so on the front, if you flip over in front of that sheet, at the very top, it says Discovery Ed. You would have already done this, except you you don't have access. They've shut this resource down and made it more private. You have to. It's kind of like Google Classroom, and everyone now wants to brand their sites and be able to set it up that way. So that's used to be you could get to all this great stuff, and now they kind of they've restricted it. But yes, ma'am. Yeah, yes, right over there. Let me grab this. Okay, so how cells become specific. So part one, the story begins. So that's where we are. And so I've snipped out little bits, little diagrams from this first little video. So I can make sure that this is right. Okay, so that should come up once I start typing. Not. Even if I try to link it, it won't let me in. Oh, it's not coming up. Yeah, come on. I should be really slow. / please work.
Yeah, so I'm just like, yeah.
Oh, let's say something about my
Yeah.
I saw my dog. Rick? The blind one? No. Walter is the middle one. Rick is the big one.
On the couch, she just attacks it.
Oh,
He never sees it coming.
When a single sperm fertilizes an egg, it creates a zygote. A single cell now contains a full set of chromosomes, half from the mother and half from the father.
All right, let me pause because that was that first one. They just jump right into it. So they're kind of, you can see how they're color coded up there. So how many different sets of DNA do you have?
30.
How many different sets of DNA do you have in each of your cells?
Like
26.
23.
Yeah, it's 23, isn't it?
Okay, so you have 23 chromosomes from each. So you had a set from each parent, and now it comes together as 46. Right. So you have then your own little individual set of 46 chromosomes, half from each parent. Okay. Here we go. And as soon as this happens, this is a good example of those internal factors because they're going to start firing off and the cell's going to start to do something.
It's still deep within a fallopian tube and has a long journey ahead, but it's ready to begin the extraordinary transformation into a complex organism made up of billions of specialized cells. Within a day of fertilization, the zygote starts to divide. This is called cleavage. Every division doubles the number of cells. Before dividing, the cell makes a copy of its chromosomes, so each cell has exactly the same DNA. After the 8-cell stage, the divide.
Pause it. Okay.
I think they already passed number 1 in the beginning. Yeah.
It is. All right, so also we call this mitosis. So I know somebody out here has thought cell division, wait a second, we didn't call it cleavage. That's just what they call this specific sequence of mitosis. Okay, it's still cell division.
Dividing cells form a dense ball called a morula. As the cells continue to multiply, they form a single layer around a central cavity. This hollow ball is called a blastula. Roughly 5 to 6 days into a human pregnancy, the blastula implants in the wall of the uterus. Now the differentiation begins. As the blastula folds in on itself, the cells migrate to form 3 layers: the ectoderm, mesoderm, and endoderm.
Okay. All right, so any questions so far? All right. So what causes the embryo cells to start to differentiate into specific types? They're going to tell you that soon. Now I want to pause for a moment because in the other classes, I'll just say, okay, now you're going to start getting specific types of tissue, and that's all I said. I want to kind of add on to that. So your mom still doesn't know she's pregnant at all. And another mammal has no clue that they're pregnant because this is so early in development within, outside of a week, a little over a week since you were conceived that this is happening. So you were this hollow ball of cells, and now it starts, and again, genes are cutting on. And when they cut on, you've got the transcription factors inside. You remember how they said something about how they kind of migrate different parts of the cell. So all of that is a cascade of events that now they're going to start to change and layer themselves. So the outside ectoderm forms all this and your nervous system. And then the endoderm becomes something, the mesoderm becomes something. So it's like they all start to move in position.
The ectoderm will develop into skin and the nervous system. The mesoderm creates almost all the internal structures in the body, including bones, muscle fibers, and blood vessels. And the endoderm forms the tissue of the digestive and respiratory organs. The human body contains trillions of cells. So how does a cell know its destiny? Each cell has exactly the same DNA, but not every gene is active in every cell, and this determines the cell's fate. For example, when it becomes a blood cell or a nerve cell. Even at this very early stage of development, it's possible to see an organism taking shape. The beginnings of a beating heart, a network of blood vessels, the extension of a limb. But it's hard to tell whether this is a frog, a chick, or a human being. That does not become obvious until later.
Okay, so what was the first one? Your fertilized egg. And then you have the same copy of what?
DNA.
DNA, and then what's the hollow ball of embryo? Blastula. Okay, so what caused all of those different cells to start to become different types?
Every gene, they have like the same DNA, but every gene is turned off.
Genes, right. Okay, so here is something that's kind of interesting. So if you have your phone, pull it out, and I want you to search something. Look up germ, like germs, germ cell tumor. And let's see if you can get a bit, like a picture of it.
You say
Germ. Because these are also called germ cells, those different layers.
germ cell tumor.
Yeah. See if you can get an image. And if you don't get a good image, like might look a little creepy, then I'll give you another way to search it. Okay, let's see if there's any other pictures, and I'll tell you to search another way. What's germ cell tumor?
I found a mixed amalgam germ cell tumor.
Okay, so those tumors have different parts in them. Okay, now search hair and teeth tumor. Also a type of germ cell tumor. At least just show, once you find it, show more.
Sorry.
I know. Nasty, huh? Same kind.
Normally
Teeth.
Oh, never mind.
Okay, so these look pretty gnarly. So what can happen in the early stage of development, the ectoderm, where is it on, where is that in the embryo, the ectoderm? The outside. Okay, so do you remember what they said those outside cells are going to differentiate into? Hair Skin.
The nerves and stuff.
Teeth, bone, nerves. So if one of those cells, y'all, has been, the gene is cut on to become one of those things, and it gets pushed inside on accident, and it still starts to develop, it becomes one of those tumors. So my friend who is a pathology assistant, he said that when they get those tumors, he said you cut into them and you can find hair, you can find bone. And he said sometimes people think, oh, these are twins, it's like parasitic twin where you like absorbed your twin. And he said most of the time it's one of those cells in development that got pushed down into the wrong place and is still trying to become what it's supposed to become. And you just don't know you have this tumor. You've had it since birth. You've had it since you were, that cell was just in there as you developed. And then eventually it gets big and, it might obstruct something, or you just notice you've got this mass and they go in there and it's one of those tumors. It's benign, so it's not, doesn't mean you're going to have cancer. But not wild. So he usually gets some of those once in a while. I've had students in the past who have had tumors before. They were benign, but it's just a weird growth and they remove it and it's just, either a big cyst-like tumor or.
So those tumors don't need a tumor. So it's just kind of.
It's just a freakish tumor. That's not weird. So that's kind of my little take on that, because it's kind of neat that that happens, but not neat for the person who has it, but it's kind of one of those freak science, kind of cool things. Okay, let me see. Got to get down to the next one, which is the blank slate. Let's see. I think you guys already. Oh, okay. I wanted to see the Bill Nye the Science Guy one.
Oh, I didn't do that. So, I feel like it's just the feeling.
Oh, and same you need not everything is
328. This is the only one we're going to do, then we'll go ahead and get into your stuff. We'll do the breakout. That's the one. Yeah. I've already had these links pulled up, y'all. Still not working. Are we balanced in your groups?
not
today.
Not today, right? Dang it, because it's just be the two of y'all. Well, hate to, are y'all okay being by yourself, or do you want to balance it? So I don't know how far you got, so I don't want to.
We didn't put anything in there.
You didn't put anything in the first lock, like the first door?
We were just like trying to like organize everything, like
Okay.
and do that.
yeah.
Then you could, y'all could oh, come on. Let's just forget it. I'm over this. Okay. All right, so what's the first one? Blank slate cell. What do we already say it was? Stem cell. Okay, plant cells have specific types of cells in there. And this is root stems and leaves for plant cells. So the next one, root stems and leaves. Okay, and the next one is 250 plus. Mm-hmm. Mm-hmm. And this is because of the specialization within the cells. Okay. So in the early stages of the development, the embryo is many blank cells that have the ability to become a different type. So many stem cells, again, have the ability to become a different type of cell, and then those are just any type of cell. So they give a list of all those at the bottom. You can just choose a few to plug in any of those in your chart down below. And then what controls the process of you becoming, or not you, a cell becoming a particular type? Genes. Yep. And then the last one is regenerative medicine in part 3. So that is a pretty fascinating field.
We don't need number 4.
Number 4, I think y'all already did part 4. That was homework. Yeah. So, and actually you did that ahead of time, so that way it will help you do the breakout. All right, so y'all want to, do you want to just kind of split up then and do it? And we'll just say whoever's, because that's the benefit of being here, you'll get the points if you, or it's actually a hall pass. Or we can do 5 points on your benchmark.
Benchmark.
Okay. All right. So whoever's first. So let me just make sure. So once you go back there, that y'all stuff or this group stuff is on top of the slide boxes. This group's is in that cabinet. Okay. And then the first door should have the 7 different. That was the cell type activity. And then you have the lock 2 and the lock 3. And those also, I think you have to use the magic pen for both of those too. So the magic pen should be with both of the things. And if you get to the point where you can't see something, let me know. Okay?
You little fishes. So when do you let them go? When they get like a little bit bigger, or
In May.
oh? So how big do you think? How big that gonna get?
Depends on how many are in the tanks. The more in the tank, the smaller they are. Okay, so this is timed because it is a breakout situation. Okay.
Isn't it, sorry.
There's a little bit. That one.
I don't know which cabinet.
It's that one. I'm pretty sure it's that one.
Is it that one? Yes.
Now, are y'all gonna work solo, Lacey and Karen? Or do you want somebody else to go with you?
Okay. Ready? I think it's this. It's in the have the face of the game. Ready? So, we need to see what it does.
Oh, there's mine.
Yeah, she's like a different flag.
I think we made some pictures in there. We all know.
Okay, let me look.
Hi, Mr. Robinson. God bless us. Please report to the main office. God bless us. Please report to the main office.
Could be. Okay.
Figure what?
Use a what?
Hey, hey, hey,
Yeah,
Is
we're
this locked one? Which one is locked? Sorry. It's special, I was like, this is.
I'll give you that last one. This one's, they have one hands. So I'll do that one. All right, well, whatever that red blood cell is, we're ready.
Exactly. This, we're doing a lot. Now put it in. Are we going to?
Alright, 12. Count down. You ready for it?
No, I don't know what this is. Yeah, but
Wow, we really?
you can tell me that. What do you want from me?
Oh my God, that's fine.
What, no, it's
Hey, what's the other one that we have to do? I don't know this. Is this the three?
1, 2, 3.
Or no, not?
No.
Same thing, I don't know.
1, 2, 3.
Oh, y'all are done? Okay, so all, you're right. You don't okay with everything? What's wrong with it? What's this? Are you ready? What is he? Oh, she said we can't see it. We have to know. We already have it. Is that a speed or? Which one is that? It's not a fly.
5. Oh,
It's a game.
We're already up there.
Gotta put it on this end. I don't know, it's kind of hard to read. Okay.
which one did you say was okay?
4.
Oh, no, wait, I was wrong. So it was K, 4 was K, right?
And 5.
5, yeah, 5 is.
Well, how many do I know?
There's part of it right there.
Right. Do you know how we can lift it
Then use your magic pen.
Oh, for this?
If you put the answers on the paper I gave you.
Yeah.
Yes, you can use your magic pen.
Can I see my pen, please?
Let's see.
Yeah.
You have to share. All right, are you up on the red blood cells now? So that's the picture you're looking for. All right, so that one is gone. Do you see anything? Yeah, you ready to go to the next one?
No, that's my house.
What is it? Is there anything here?
You saw those sacrifices from, like the workers.
Okay, so. Shut up. Good job. What? Oh, can we put our clothes on? Or no? Okay. Which one's number 2?
No, ours should be up.
Are you have 8 minutes?
Is this one number 2? Oh.
That part's on.
What? Yeah.
In that order. Cause we thought ours was the worst.
You'll know when you're done. What's it tell you?
Have you already done your names? Yeah.
You just did it. We're done. All right, they won. 6 minutes left.
Wait.
What number comes first?
Which number comes first?
All right, let's go. And then, let me give you a folder. We'll put all that inside. A little plastic sheet.
No, he's. That makes no sense.
We'll just use this.
Backwards?
All right, where are y'all?
All right, what did you say it was?
I don't, like, it says it has to spell something. So which one?
What number 2?
Oh, not number 2.
Number 2
This
is
is
those.
number 2.
Let's see what page. And OG.
And then this one, right? Oh, wow. And so your name's a little confused. Can I just do it? No. You're such a bother, you know that? Don't.
Y'all A-M-A-R-E.
Okay.
Um,
Just put everything in here. Just dump everything. Okay, you put the pens in there, everything. They realize I was not too.
All right. So, you're gonna need, gracious, a yellow, I use a yellow, blue, red, and, something else, purple or something, but I always use yellow. yellow and blue, but then it doesn't matter. So you need like 4 colors. And we're going to move to your notebook and kind of switch gears to talk about, in the same vein, about cell membranes and signaling and these different factors that cause cells to do something. What, Waverly? Pens, color pencils, markers, yep, any of that.
Colors again?
I'm going to use yellow and blue for sure, and then the other two, it doesn't matter.
Where would you like this?
Thank you. Eventually, we'll be able to use the breakout boxes. And
Can I actually ask if you can get the notes on YouTube?
you go on YouTube, and it's my channel. So Thornhill Biology, you'll come up. Let's see Thornhill Biology, and it'll come up. So right now it's just taping a blank screen. So all she has to do is rewind and go to the part that she needs. So, all right. So everybody has the new packet, and this is part 2. We will do A, B, and part of number 1, and we'll do part of number 2 today. So we're going to really kind of move through, but please ask questions as you go. We're going to draw them. Yeah, we're going to put them in your notebook. So it's been a little while, so we need to go back there.
Is highlighters okay?
Mm-hmm. Yep. Not really sure why that's there. Okay, so this is still Unit 2. Cell transport. Okay, so the last time that we talked about the cell membrane, tell me what you remember. Okay.
You said it's very flexible rather than the cell wall.
All right, so it's way more flexible than cell wall. Okay, good. What else, y'all? Every cell has one. What is its function? And what does it do for the cell?
It protects the organoids that has inside of it and transport things.
Okay, so it does offer a layer of protection just because it keeps all the cytoplasm inside. Okay, transport.
It helps with like what goes in and out of the cell.
Mm-hmm. So you already remember a lot. So that's the first part. What we're going to focus on is the structure, which is a little bit more detailed, and then we're going to be reminded of everything that you already said. So the cell membrane, there's this theme of structure reflecting function. Structure or form and function, reflecting function. You're going to see this all through biology.
Will this be notes? Or like on the packet, do we write this?
I mean, you write wherever you want to, just all notes. And so that's not listed there. But you clearly saw it for the different types of cells. So the shape and what that means is shape, composition, and design of whatever the structure is. Indicates its job. Okay, so a non-cell example would be a hammer versus a screwdriver. So a hammer, you know that it really can be used as an extension of your arm it could be a lever. Oh, it just cut off. Great. It can be used as a lever. It could be used to pound stuff. I don't know what this thing is doing. And then we have different types of screwdrivers. So what would be a structure and function? Another example that's not a biological example. So like, can you think of the design of something tells you what it does? Or the design helps it do a particular job?
A wind blower?
Okay, so how so?
Because it like it pushes the things, like pushes things out, like blows. It consumes air to like to push the things through. I think.
Okay, so how about, so the design of it has wheels, it has the blade, so it has so everything. Okay, what else? What's up? What?
A car.
So now tell me some examples of what you just did, the different types of cells that you looked at, versus why is their design a certain way? Or how does their design help them to do what they're supposed to do for the body?
To help it carry out certain functions.
So structure and function, we're going to find out is even when we look at everything as simple as your hand, why you have an opposable thumb and your toes aren't. You don't have an opposable big toe, but you do have an opposable thumb for a reason. And we know if we x-ray the flipper of a seal, you know, outside it looks like a flipper. And so why does ours not look like a flipper, even though we have the same bones as they do?
We evolved to use our fingers the way we have to.
So through the process of evolution, but it's because ultimately, where do they live and where do we not live?
Underwater.
They evolved from a four-legged animal, moved into the ocean. So at some point, mutation started this series of mutations and evolution, allowed them to their outside to be more of a swimmer type appendage than a grasper. So that's structure and function. So when we look at the cell membrane, I'm not going to read, everybody get what's on top. All right, so the structure and function. So when we look at the cell membrane, it's designed in a particular way to do a particular job. So structure and function is design of a structure, is composition, reflects its job. Word is in. So a nerve cell, we have these dendrites. This is the cell body. All the cell parts that you've learned would be right inside of there. We have the axon and axon terminals. So why is that designed that way and a red blood cell looks like a flexible disc? Two different jobs. So you need to have a whole different design. So that's the same reason that your stomach, empty, if we do a cross section, looks like this. A mitochondria looks like this. And the inside of your intestine looks like that. Oh my goodness. Why do they all have these little folds inside?
So they all have to.
Sorry, you go ahead.
Oh, because like they all have to like hold stuff inside of it, and then they have to get out all the like nutrients and stuff.
Okay. Well, you're starting to. You have to kind of talk it out, because if this is empty, and the mitochondria has the same setup with all these folds, opposed to your stomach looking like this, your intestine looking like this, and the mitochondria looking like that.
So because they all work hand in hand together.
They work what?
They all like work hand in hand together. Is that why they all work on this?
Not really. I mean, they do, but it's not even the connection between the two.
That was the same DNA for seven different functions.
Why would you want to have all this space
And you can put more stuff inside.
and to do more work at the same time? So this is an example of structure and function of an increase. It's called surface area. If I can increase all the surface space, I can digest more food at one time. I can make more ATP at one time. So that's an example of structure and function with the increase of surface area. Okay, so it's interesting. You're going to see, now it so happens that this is important. So this stuff, this mitochondria can get its glucose in its parts, just so it can undergo cellular respiration. But why I even say that is because the cell membrane is flexible and is designed out of what it's made of to do a particular job. So structure of the cell membrane. We're going to draw it out, and that's what these things have been hanging here forever. Okay, ever since first and second period did this little skit as the cell membrane. So structure of cell membrane. Now we also know that it's called plasma membrane, so sometimes people will use that instead. Okay, so what do you remember? What is it made of? What two major organic molecules? Lipid. So it's phospholipid and something else. Protein. All right, so let's draw the phospholipid first. So the first thing we're going to do is when I draw it, leave some space up here so that way we can write something and label things. So I'm going to draw, this will be in yellow. Okay, I'm just outlining it. So let's say it's going to look like that. And then there's going to be one on the bottom because it's a phospholipid bilayer. So what does bi mean? What number? 2. 2. So there's 2 layers of lipid. So I'm kind of cheating by putting this at the bottom. So technically it would look like this. It'd be like a balloon with 2 tails, 2 strings that you're holding. And then there happened to be one on the bottom this way. So I'm going to draw 3 of them in a row. So here's one. That way. And then here's one. All right, so we're making it very 2-dimensional looking, but we know that this would extend out this way and you would see several of them. So if we were to bunch these up, it would be like holding a bunch of balloons as you're walking. So if you're holding on to the strings, what are the balloons doing? Are they staying perfectly still? So that's true. As part of the structure of these lipids are shifting. Your cell membranes, those little parts are actually moving. All right, so here's our lipid bilayer. So let's label phospholipid bilayer here. Okay, so phospho. All right, so we know that there is a head part and 2 fatty acid, the little tails. Okay, so lipids have fatty acids, and so you've got those little chains, and then you've got the little head at the top. And that's what this is a model of. That's really what one of them looks like. Kieran and Blake Inhabit made that last year. Okay, all right. So now I'm grabbing the blue, and this is going to be the protein. So you have phospholipid, and then we have different types of proteins. So the first protein we're going to do is a transport protein. And so it looks like that. And there's different types of these, all transporting and moving different stuff in the cell. But it has a gap in between. So think of it as like a checkout line at a store. So this is transport protein. Or sometimes people call them carrier proteins. So this is going to be the outside of the cell, and this is the inside of the cell. So we've been doing this. when you drew your, for your microscope lab, you drew a line for the cell membrane. We're taking this, and this is what it actually looks like. So we're really going to go in, like with an electron microscope, and zero in and see the composition of the cell membrane. All right, so let's do like 2 more of those lipids. Okay. Then we're going to draw a different kind of protein. Here we go. Okay. So this kind of protein is going to be kind of stuck in the membrane, and it's going to have different shapes, and they're going to be part of that cell signaling. So we said that there's external factors that can influence a cell's ability to do its job. So we're going to do actually a receptor protein here. So we're going to make it look kind of jagged, and it doesn't pass all the way through. And then we'll put a couple things down here. Okay. All right, so this one we're going to label a receptor protein. And then I'm going to just put a little star because I'm going to come out here and write down what it does. That way we have more space up top. Okay, so how this works, what are the different what do we call the chemicals that your endocrine system makes, and your which pancreas is part of that controls your development or your digestion, sexual development? We usually think of, what do we call those chemicals in general? Insulin is an example. Hormones. So hormones would be a signal. They are chemical signals. And how they operate is it's not like a magic wand that just goes bling, and then it's like all of a sudden the cell just does what it's supposed to. What happens is they're put into the bloodstream, and they have to dock onto the right protein, and that sends a signal, and that tells the cell what to do. It's pretty crazy. So that signal, that hormone, might actually put on a gene. It might tell the cell to do something. So we're going to say that a receptor protein receives specific signals. We'll put chemical signals that cause a change in a cell. Oops. Right, so then we're going to take another color, which doesn't matter what, and we're going to draw one of those signals. So this is a hormone, maybe like insulin. And that would be the chemical signals.
So the red thing is the hormone.
Mm-hmm. Which would be like insulin. So let's say that this is, you have to have glucose. You have to have sugar coming into your cell so your mitochondria can make ATP. So when you have insulin, your pancreas makes that insulin. You've eaten sugar. So I use second period, for example. If you got grab and go, boy, there's some Pop-Tarts. Okay, those Pop-Tarts loaded with carbs. So your stomach and your digestive system is breaking it down and puts it into your bloodstream, y'all, that sugar is going to stay out in your bloodstream unless a hormone tells these cells, hey, you've got to take this stuff up. It doesn't just automatically come in. It has to be told to take it in. So what happens if you're diabetic?
It don't need it.
So where does all the glucose stay?
It's just like goes to your bloodstream.
It just stays out in your bloodstream. And then when your kidneys are filtering your blood, you pee out that sugar. So when that little test that you do at the doctor, and they dip that little stick in there and they say, oh my goodness, there's glucose in your urine, that's how they know that you could be a diabetic. So that's why it's important, if I'm a diabetic, if I'm type 1, somehow either my pancreas is not producing enough of that insulin to actually get out here to tell these cells what to do. So I need something to help me out. And so that's why people take it for that reason. All right, so let's do one more protein, and let's just do, we'll make it look kind of like it doesn't have a specific shape, but this one is going to help with cell recognition, cell ID. So we have proteins that help with signaling, but we also have proteins that tell your immune system that, hey, these are my cells and not someone else's cells. Okay, so we get a third color, it doesn't matter what it is, and we're going to make a little ID tag on these. And these are carbs, that's why I'm trying to make little rings. It doesn't matter, you can make one that looks like a Y, you can make one that looks like a W, a V, it doesn't matter, because your ID tags, because of your DNA is unique, your ID tags on your cell membranes marking your cells as yours are unique. It's kind of a cool thing. Okay. So I'm gonna, this is a carb ID tag. So this is how your immune system knows when stuff comes into your body, it knows it's not yours. So what if a virus came into your body and it doesn't have your ID tag? What is your immune system going to do?
Send out, and send someone out to get them.
Yeah, so what kind of cells are going to do that? White blood cells. Okay, so what if you, like, all right, Selena Gomez, I used her as an example before of organ donation. So she needed a kidney, right? Don't know that story? Mm-hmm. Okay. None of her family members were matches enough. So, you know, in an interview, then she comes out after the fact and she says, her roommate, her best friend, happened to be a perfect match. Crazy set of events. So what's interesting is these ID tags, even though she's a close match to her, if Selena Gomez and any other person who's received a foreign organ or part, you know, in their body, they have to take anti-rejection drugs because your immune system is still like, oh, no, that is not your organ. That's not your organ. Those aren't your ID tags. So what that drug does is it says, okay, ignore that. And your body doesn't reject it.
My aunt just got a kidney transplant. Like, she's been on dialysis for like 28 years. So, like, she hasn't been able to pee. So, like, now she got a,
There's one dying.
Okay, yeah, fish them out. Yep, thank you.
so, like, now, like, she has it and now she pays it.
So that's where it's pretty remarkable that we have that, but she's going to take those, you know, that, ask her about that and see what she has to do now that she has a foreign, you know, she has an organ that's not hers.
So
What does she have
the
to do?
doctor said that the kidney's like working so well, it's like it was actually hers.
ask her if she has to, what kind of medication she has to take for that. Or if they were, what does she have to do? Because I'm curious. So the ID tag identifies your cells as self, okay? And no one else's. So this is where the immune system, how it knows what's not self. Okay, sometimes there are autoimmune diseases, and that means your body is attacking itself. Lupus, I think fibromyalgia is kind of like that also. There's different where your body is like, oh no, that's not you, and it is you. So sometimes that does happen. I hope not, but it can happen. All right, so that's how it works. Now, as far as the flexibility, this whole setup, if we were to look down on top of it, it would look like a mosaic. So like this floor, all these would be the heads of the lipids. All these orange and blue boxes will be the proteins. And so it looks like a mosaic. So this is called the fluid mosaic model. Of the cell membrane. Now, you guys already said that it's selective. So what type of things should be able to get through and squeeze through and get through these lipids? What type of smaller kind of molecules do you think are allowed? Oxygen. What else? Carbon dioxide. Water. Those things. So what we find is that if it's a small enough molecule, it should be able to squeeze through those cell membranes. We'll go this way. If it's big, it may or may not be able to get through, just through the membrane itself. So there's some different strategies. Sometimes substances have to be moved through those. So that's what transport is. So passive transport versus active transport is the whole idea of moving and maintaining homeostasis. And that's what the cell membrane's job is. Okay. Are there any questions so far? You want to go back over anything as far as the structure of the cell membrane?
The transport it's active, right?
It could be both. Yeah. But it's going to transport substances either a little bit quicker because it's more like a pump, or it leaves a little gap so substances can get in a little bit faster. Okay. All right. So I think that is it for that part. Yes. Okay. So let's look at in your packet, and you'll need your laptop real quick. And what we don't get done, we'll just make it for homework, but I think we'll be able to be part of this. Let's see. Okay. So we turn to the third sheet, I think it is. Yeah. Zoom out, too close. All right, there's two sides. One thing, I love this, the selectively permeable nature of the cell membrane is demonstrated on this site. They've taken this down, and it was so good. So it's still here, left over, so ignore this side. Come over to where it says signals. So here's another example of how different signals can cause different changes inside cells. So remember those proteins, and we're going to be able to react certain ways to different chemical signals. So go to Google, and we're going to do the first line together. And then we're going to see how far we can get. Okay, so once you're there, it's easier than doing what I was doing earlier, trying to type in a URL. So we're just going to search the topic and it should come right up. Okay, so dropping signals, not drooping. Learn genetics. And it should be the very first one that comes up. Okay. So if you're interested in this part of biology and genetics, this is a really cool site to visit. There are all kinds of neat stuff. Okay, and you'll have to click on this little puzzle piece to make the flash work and allow. Okay, so we're still talking about specialized cells, and they all have different proteins that react to certain external cues. So in this case, we're going to expose it to 4 different things. So at the top of your paper, flip back. These are the different external stimuli that you're going to expose them to. The different cell types are down here on the side. So we're going to click on muscle first and see how it reacts. So we're going to click there. And there are your 4 different stimuli. So when we drag one over, it's going to tell us or show us what happens. All right, so let's do light and see if light affects your muscle cells, if you just shine light on them.
This muscle cell does not respond to light.
Everybody's going to be like in stereo. All right, so no response. So that means light has no effect on your muscle cells whatsoever. So we're going to go on your paper and we're going to put no response under light for muscle. And then you keep going. So we'll do the next one. So nitric oxide. This is laughing gas, like at the dentist. Okay. So if we give yourself some laughing gas, we'll see what happens.
Nitric oxide is causing these muscle cells to relax.
Makes sense, doesn't it? Okay. Relaxes.
Do we have to write 2 sentences, or do we
I just kind of put no response, and I just put relaxes them. Okay. And then cytokine, which is a growth factor. So it's a hormone.
This muscle cell is responding to a cytokine by dividing to make 2 cells.
So it divides to make more cells?
So it causes them to divide. Yep. Stimulates. To divide, or you can put, cause cell division, or however you want to word it. And then the last one, so it just says generic hormone. I think it's, let me see.
In response to a hormone, this muscle cell is taking up nutrients from a nearby
It's
blood
insulin.
vessel.
That's what it is. So it's going to help it to end. Yep.
So, takes nutrients?
Mm-hmm. Go too big. Nutrients. Okay. Let's see. We only have 6 minutes, so we'll see how many you can do in the next 3 or so. See if you can get through. And then whatever you don't get done, we're just going to finish for homework. And I'll just leave it at that. Do the same thing. I've got those. Alright, I'm gonna go ahead and you're on your way back. Hey. Oh, yeah, you can come in the back door, it's fine. Oh, yeah, you went in that one. Yeah, I was gonna look out. I hope it's unlocked. I'm just gonna try to, yeah. Remember Tiffany George?
Yeah.
She used to call that the underground railroad. All the time. She'd come through there, she's like, underground railroad. Oh, my God.
Cells damage.
Did you put program cell on?
What?
You put program cell on? Does anybody have any questions so far? Let's see, we've got 3 minutes to the bell, so
I
if
just
you can
put
get
cell
it,
death.
you might be able to get maybe one more line done before, well, 2 minutes left.
Can we start to pack up?
We got, 8.
Yeah, that's okay. Now we can get it. What we're doing.
So, I mean, we could just, I could just, it was more.
Well, I don't know. Why?
I want y'all to come back and, y'all can leave it at, this is B day today. I have. And yes, we are here. Y'all have a great weekend.
Or you can go.
Yeah, so I have this rich every other day. So this is, they have year, this is the honors biology, that's up against my bio, up against my AP, and that's an elective. This year required. Yeah. So,
And how they flop it, maybe they.
We have a lot of classes now that are AAB and mainly just, well, it's mainly to accommodate AP biology and, AP classes just because we offer, AP biology, not this year, we did last year, but AP harmony, AP cal,
Human geo,
world,
psychology,
psychology, US, government, arts,
English,
this year, A push, US history. Then we have 2 Englishes, so we offer 11 different APs in our school.
You know, I also opened last year in high school. I was doing my assistant principal, like, internship thing, and we were trying to, whenever we were scheduling kids, we had trouble scheduling, getting kids all the AP classes they wanted, because only having 8 slots. So, like, how many classes do we need to have a year? Is it like 8?
They'll take more, they will, because let's see, if you have, some kids they'll have, their first 2 classes are, they'll have 4 for that, they'll have 2 and 2, and then they'll have year long, so it'll be 8. And some will be more.
Wow.
So, not many anymore, but sometimes they're opposite each other. So then, you still have those 2 that are going to be just.
And then the whole school, like A and B, is just.
It's school, well, it's 5 students, so they do certain students. You have world history, you have AP world, no, you have your book, your book. So what so that's how they had to give me a class opposite my AP class. And that's how they created a small little class of 8 girls. Honors biology for the year.
Gotcha.
So it won't work if it's not honors, mainly because lack of self-discipline. You can imagine every other day in a regular class because they would hate it.
Yeah.
They wouldn't keep up with their assignments. They are they're very independent, so I don't have to worry too much. Yeah. Let me cut this off. I was videotaping this for this